is Aidan Husseinov. I'm biotech analyst here at Ladenbrug Thalman. And our next guest is Allarity Therapeutics and CEO James Cullum. Welcome, James.
Thanks very much for the introduction, Aidan, and thank you to Ladenbrug for the opportunity to present a little bit about our company, and thanks to the audience too. We'll try to get through this and save enough time for some Q&A. My name's James Cullum. I am CEO at Allarity Therapeutics. I'm joined today by two of my colleagues sitting on this side of the room. Thomas Jensen, who's one of our company founders from Denmark and currently serves as an SVP to the company in investor relations, and Dr. Marie Faux, who is our CMO. So you may hear from them if we get some tough questions. Allarity Therapeutics is a clinical stage oncology therapeutic development company. We are working diligently a way to realize true personalized cancer care, which can only be achieved by having robust, clinically validated companion diagnostics, and we have a platform that is just that. We think it's quite unique and sets the company apart, and we'll talk about that today. We are publicly listed on NASDAQ. The ticker is ALLR, and this deck that you're going to see today is excerpted from our larger company deck, which is available for download from the company website. That's our legal statement and disclaimers. Quick snapshot about the company. We originally were founded and operated for many years near Copenhagen, Denmark. We still have an R&D facility there where about half our team is based. We migrated the company executive management and the company itself to list on the US NASDAQ last December, underpinned by an initial $20 million capital raise from 3i Fund here in New York City. We're developing a robust pipeline of 3 clinical stage oncology candidates. They're all former big pharma assets. They're all, in our minds, substantially de-risked, which is part of our business model. We'll talk about that in a moment. And for all of our assets, the secret sauce, so to speak, is our development of a DRP companion diagnostic for each of our assets using our proprietary and very well-developed DRP platform technology, and we'll talk a little bit about that. This is a short slide on our pipeline. You see at the top, we're making good drugs better with DRP. This is an absolute central piece of our strategy. We're not looking for new lead molecules. We're not doing anything preclinical. We're not doing first-in-human studies. The assets that we've acquired are, as we mentioned, all past phase 1, all safe, well-tolerated, all have shown prior signs of efficacy, including one of our assets, Ladenbrug, which was at the end of phase 1 when we picked it up. So we have gone out of our way to choose assets that we think are quite far along the development pipeline and we can make better by applying a DRP, drug-specific companion diagnostic. Get to the next slide here. Personalized therapy, of course, as we all know, it requires robust, validated, predictive diagnostics that actually work in real patients, not just in cell lines, to identify the responder patients and select out the non-responder patients. That's the holy grail of personalized medicine. Our approach to do it is through the DRP. What is DRP? It's a unique transcriptomic-based bioanalytic platform. It's sort of the secret sauce, the magic box of the company. How it differs right from the get-go is we assume we know nothing about what drives tumor response or resistance to a given cancer drug. We let the tumor tell us what's important. You see a graphic on the left. Cancer is extremely complex. Because cancer is extremely complex, fairly simple biomarker approaches to select patients largely have not worked over the past 20 years. They're too simplistic. They don't capture what's unknown about what's driving tumor to respond or not respond to a drug. So we start off every time with our system of assuming we don't know anything. We take the entire set, 25,000 or so expressed genes in cell line panels, and we analyze initially which of those are correlated to responsiveness and which are correlated to resistance to a drug. But we know for 30 years of cancer drug development that what occurs in cell lines most often doesn't translate into the clinic with real patients, because real patients have tumors inside a very complex environment. So what we do in our system that's quite unique is we then pass our initial signature, if you will, which typically you see on the slide. Typically we have 50 to 400 gene biomarkers in a specific a signature of response or resistance that we develop. We then pass that through what we call a clinical relevance filter to be sure it translates to the clinic. Our clinical relevance filter was created over 15 years or so by obtaining and analyzing over 3,000 human biopsy samples from actual human clinical trials, spanning a broad range of cancer drug types, spanning a broad range of cancer indications. So our initial signature goes through the filter. When it comes out the other end, what you essentially see here in color spectrum form is a putative DRP diagnostic signature where, and again, from 40 to 50 to 500 genes. Green is a DRP score, which is a composite score of all of that. Green is a high match. A tumor would have a high match to likely respond to a drug. Red, green is go, red is stop. Red means that you have a lot of the resistance genes and you don't have a lot of the response genes. So we have a tool that lets us both enrich for responders and to exclude non-responders. Before we can actually use this on real humans in clinical trials or ultimately on the market, once we get an FDA or regulatory approval, we have to do one more important step that most competitive approaches in the field don't do. We have to convince ourselves that this actually would have predicted responder patients in actual human clinical trials. So what we do before we use it, we take our DRP, we do a retrospective blinded analysis, we obtain biopsy materials from a clinical trial of that drug or hopefully multiple prior clinical trials of the drug, which we'll show you some examples. We get those biopsy samples or the transcriptomic data from the biopsy samples blinded as to the patient response, any patient information. We make predictions with the DRP, and once we prove that we, in fact, would have selected most of the responders and excluded most non-responders, we take it into the clinic. Now we have a companion diagnostic to use with real patients. At that point, it's a simple matter of we get a patient biopsy sample from a potential patient that wants to enroll in one of our studies. We look at the expression of the relevant genes in our signature. We match it up to the DRP, and again, you see the color spectrum simplifies it, but we're looking for the green. We're looking for the higher responders, excluding the red ones. And in the middle, you get some yellow folks. You see on the right-hand side, they might be eligible. We can move the cutoff of the DRP. We typically set it at 50%, a score of 50%, which we have shown over the years in many, many studies captures most of the responders and excludes the non-responders. Again, DRP is unique, we believe, and best in class because it's been developed over 15 years by the company, extensively studied. You see here 47 clinical trials. Most of them in the bottom were retrospective blinded studies. We've published a lot of this work. It's peer-reviewed, published, it's patented. Up on the top, you see some of the prospective clinical trials we have run or are running now to show the power of the DRP. So this platform, broadly applicable, and we could, for any program that we bring in, we develop a drug-specific companion diagnostic. We can also out-partner this to advance other people's pipelines, et cetera. Last note of this slide over here in red, 2 to 5 increase. Why DRP? Why do we care? Because it robustly has shown that it can double to quintuple whatever the relevant patient benefit is that you're looking at, whether it's PFS, overall survival, et cetera. Now to our programs. This slide's very busy. Please read it in your own leisure, and you can find this on our website. We're not going to hit every bullet point. Davitinib is a unique pan-TKI that was developed extensively all the way through phase 3 clinical trials by Novartis. We picked the drug up from Novartis some years ago for a number of reasons, including that it had in at least 5 different indications in the hands of Novartis shown exciting efficacy. Ultimately, the lead program was third-line metastatic renal cell carcinoma. We initially were developing the drug in that area. We changed direction, as we announced publicly, due to some regulatory hurdles on that. We could talk a little bit more about that. But the compound is safe. It's got a number of indications where it has efficacy. And we, down at the last bullet point here, you look at another drug that we consider somewhat similar in terms of the development path, which is Lenvima. Eisai is very successful, or Lenvatinib. Eisai is very successful pan-TKI, which has now gone on in a short number of years to generate $2 billion of sales, largely being driven in combination therapy setting with Keytruda, PD-1 setting. We really look at that as an example of where this molecule can go. This is a quick snapshot. I want to move through of the work we did to validate that the Davitinib-specific DRP actually works. And to say the long and short here, we obtained all the biopsy materials from Novartis' phase 2 and phase 3 study. We retrospective blinded, analyzed all those. We proved that we would have picked most of the responder patients. And you see on the right-hand side, what was the benefit? We didn't quite double, but not far off of doubling the patient benefit in OS. I'm going to move through this just for time's sake, but one immediate opportunity that we recently announced to the market for this drug is to develop it in combination with our PARP inhibitor, Stenoprib. We did announce to the market that the company is making a pipeline strategy refocused towards combination therapies. That's to try to catch up with where the market already is. When you look at the field of standard of care in cancer, already the actual existing standard of cares have shifted mostly to combos. A lot of those are with immune checkpoint inhibitors, and we're coming current to the time trying to pursue programs moving forward that we can really maximize patient benefit and sort of compete and drive value. This is just re-saying what I just said, so I'll move through this, that we've thought, along with everyone else in the field, what might we do with immune checkpoint inhibitors, because they're just showing such great synergistic effect. And that's, when we talk about combination therapies, we're looking for synergistic effect. We're not doing these things because we're going to have additive effect, get 8% response rate, add it to an 8% response rate. You're looking to go from 8% to maybe 50%, and on top of that, we can make that even better with DRP. Our second program that I want to talk about here, just mentioned Stenoprib. We acquired from Eisai, another de-risked former big pharma asset. We picked this asset up because in its phase 1 study, it showed a couple of partial responders. So we saw promising sign of efficacy early on. Once again, we obtained biopsy materials from Eisai, and were able to show that, develop and validate our Stenoprib-specific companion diagnostic, that we would have picked those responders and we would have excluded the non-responders. This asset is currently in an ongoing phase 2 study with US and EU sites as monotherapy in ovarian cancer. And there's some information in the deck I'll skip for now about when the data readout's going to be on that. But we have an ongoing study, and we've announced that we're going to soon be starting our combination study for this asset. This again is just an example of a DRP validation, and because we were looking at biopsies from a phase 1 study, we didn't have a large number of patients. I think there were 15, 16 biopsies we looked at, but it statistically was strong enough to validate this DRP. Of course, we like to get phase 2 or even phase 3 materials because we get a lot more. And you can see on the left-hand side that there's a 50% cutoff, and we selected the responders. We exclude most of the non-responders, and some information about how much we improved, a little bit more than doubled here, the response rate. I'll quickly say on this slide, there's a lot here. We're excited about this molecule. We think it's potentially best in class. The PARP inhibitor space is very competitive, but this molecule has some very interesting, unique characteristics. One is it's a dual inhibitor of PARP and tankerase. Tankerase is a telomerase maintenance enzyme that has a lot of importance and impact into the Wnt signaling pathways. And that's just a fancy way of saying that it gives us another potential mechanism of tumor cell death. We have 2 ways we think that we're inducing death in these cells. And down at the bottom, you just see a summary of the check marks of places that we are differentiated against competition. One is we have very low to no myelotoxicity, which plagues the class. We cross the blood-brain barrier, which is always a tongue twister, et cetera. So we think we're well positioned there as long as we can get to positive clinical data readouts. Last but not least, Exempra, another former, or third lead asset, if you want to call it. This is a former Bristol-Myers Squibb asset that actually was approved and came onto the market and still on sale in the United States. It's a microtubule inhibitor. We picked this asset up because, number one, we had already created a pre-validated Exempra-specific DRP, so we were confident we could use that to improve response rates. Number two, we acquired the European rights because Bristol Myers never succeeded in getting the drug approved there because of cost benefit, the difference cost benefit ratio in socialized medicine opposed to here. So we were confident, and we are confident, we could get the drug approved in Europe, which of course would be huge for a drug that's limited right now to the U.S. to open up a market like that, and also sets an interesting future paradigm for us where we could acquire assets like that failed to get approved in another jurisdiction based on cost benefit, and DRP can move the needle. I'm wary of my time. I want to leave some time for Q&A, so we'll speed up a little bit here. Another nice graph here with our color spectrum, and you can see again we can select the responders, select out the non-responders, and we can make a meaningful impact on patient benefit. Quick pipeline development slide here. Again, you can look more at this, but what's in orange down, not orange, yellow in the bottom are ongoing active current phase 2 studies. The ovarian cancer study for stenoprib is monotherapy, and then down at the bottom, that's the Exemplar study, which is for second-line metastatic breast cancer in Europe. The rest of what's on here are studies that we intend to run or are considering running. The one, I don't have a pointer, but if you look in the middle, there's a white box that says phase 1B. We've announced we're doing that study. We're ramping up to do that first combination study, which again will be in second line or later ovarian. And then the last thing here to do, because we're speed dancing, but I got to make note, the gray bars you see there, that represents pediatric development programs being done by our partner, OncoHeroes, which recently had a wonderful announcement earlier this week, or maybe late last week, that they had received a rare pediatric disease designation for their program from the US FDA. We consider that very exciting because it's our molecule, and they will be developing it with, again, with DRP to get the results to be even better. That's in pediatric osteosarcoma for davitinib. Let's pop through this quickly. Our leadership team, we have almost half of us here in the room. The main thing I want to say about this is that our two company co-founders, Thomas Jensen, who's here, and up in the right-hand corner, Steve Knudsen, who also the two developed the DRP. They've been with this company from the get-go, which is close to 20 years now. So we have some consistency. We've got all that brain power and all that experience, and we're still very excited about what we can do here. Advisory board, you can see the names. Many of you probably know these folks. Just want to point out up in the left corner, Dr. Dan Van Hoff is another longtime prominent believer in the DRP. I've been with the company 9 years. Dan was here on our advisory board before me. So we have some KOLs that continue to believe in the potential of what we can do. Board of directors, it's another one I'm going to hit quickly. I want to look up to the right-hand corner there. Dr. David Roth at Syros is our latest addition. David is fantastically experienced oncologist, both with clinical oncology experience, but also cancer therapeutic development. So he's bringing a unique skill set to us there. And just this week, we announced the addition of our new member, Jerry McLaughlin, who's the current CEO at Life Sciences. And Jerry is sort of a serial CEO in the therapeutic development space. So he's going to bring us that big mind and help us there. Upcoming milestones to create significant value creation for these programs. We're looking for data readouts with value inflection that we can hit within the next 15 to 20 months. For davitinib, it will be the initiation of this new combination study. The first step of that study will be a phase 1B combination dosing study, but we're designing it in a way that we're going to see hopefully early efficacy signals. We hope we will get to, we're anticipating a data readout on that one before the end of next year. Stenoprib, the same program, and then the expected data readout on our current phase 2 monotherapy study, which is at or around the end of this year. And then last but not least, Exemplar, we've reported out before to the market, anticipated interim data readout on that phase 2 study in metastatic breast cancer at or around the end of first quarter of next year. With that, I will stop talking. 5 minutes for Q&A. Thank you very much.
Companies and some others are taking projects, pharmaceutical development companies. So just for the audience to better understand what DRP, what are the comps for DRP? Is it more like Oncotype DX? Is it more like Foundation Medicine? Is it more like 23andMe? So just to, for the audience, just to better understand what that is.
Great. I love that question. It's great. It looks more of the examples that you mentioned, like what Oncotype DX is when Genomic Health came out with Oncotype DX, a complex gene signature that is predictive of something. And what it was predictive of, I think, was relapse of a particular cancer, not of therapeutic benefit. So we are similar to that in that these are complex signatures. They are not simple signatures. And an example of something more simple is, pick any, a single point gene mutation you wanted to out of Foundation's NGS panel, right?
Right.
So we are more similar, I would say, to Oncotype DX. Another example is MammaPrint from Agendia. Thomas, did you want to add something? Yes, thank you very much for that, Thomas. I didn't point it out on the slide, but there's a footnote on our Davitinib validation slide that we were comparing it against the gold standard that Novartis compared it to, which is sorafenib, Bayer's pan-TKI, and our data show exactly what we expected. We do not select responders for sorafenib. We only select it for Davitinib. That's the level of specificity.
Nice. Okay. Yeah, this is helpful. So you mentioned that you're planning to have a combination study with Pembro in the future, and you have some unpublished data. I don't know how much you can share for that data, the preclinical data, and also that the DRP predictive score would that be in combination with Pembro as well? So would that be predictive responders for Pembro as well?
Many good questions. I can see you've been doing your homework, Aiden. So we, like everyone else in this field, have been looking long and hard at whether we might have potential synergistic combination programs we could run with immune checkpoint inhibitors, not by any means limited to Pembro, right? There are many other options in there, and there are other immune checkpoint agents like CTLA-4 inhibitors, which could be interesting. And obviously, they've transformed the field in terms of, at least in some cancers and settings, just transform the standard of care because of the synergy. So we have been and still are looking closely at how we could combine one of our assets with Pembro or some other agent. As far as the DRP, our current thinking has been, and Marie, my Chief Medical Officer, can jump in on this one, probably in terms of using DRP to enrich, we would probably do it for Davitinib, for example, not necessarily for Pembro. We don't currently have really a validated DRP for a PD-1, although we've done some work in that area. So that's sort of the thinking. There was one other part of your question I forgot.
I'm most interested in predictive in designing a DRP for Pembro, maybe even outside of your specific trial. If it's other than PD-L1 expression, how we can predict the value of Pembro. Is it possible to develop DRP for Pembro just to understand which patient would be more responsive to it?
So again, I mentioned that we have done some early work on developing a DRP for a PD-1. I don't know if it was Pembro. And Thomas, you might know this better than I do if you know the answer. We've looked at it, and I think we were able to get some early comfort that we can have a predictive DRP there, but we haven't gone down a normal path of validating and really testing that out.
Understood. So, but do you think that the company that provides Pembro, do you think they will provide the drug for the trial?
I doubt that any of the big pharmas will do it, just because any combinations that you can possibly think of are competitive to everything big pharma's doing, right? So now you're in a situation where if you could get Pembro or anything else as current standard of care, then you can get it for free to do that kind of study. If not, you're in a position where you have to purchase it, which obviously, or collaborate with the NCI and try to get it that way. So that is actually one of the reasons why we selected our first combination program to be with two drugs we own already.
Right.
Simplifies it.
Okay. You also, James, mentioned that there's a partnering opportunity or out licensing opportunity for the DRP asset, for DRP for specific drugs. So could that be a source of your non-dilutive financing at all in the future?
Great question. It definitely could be. We've always been open to it as long as that outsourced collaboration isn't competitive to something we're doing. I think we take it opportunistically, and yes, it could be a source to pull in some extra funding if we find the right partner.
Okay, great. All right, thank you. Thank you very much.
Thanks very much.
Thank you.